Good morning, children. So welcome back again. So now we are moving on to our next type, that is belongs to the group Bacillariophyceae or diatom. Here we have to study only one species, that is pinnularia. Okay. So we finished chlorophycean, we finished xanthophyceae. Now we are moving on to Bacillariophyceae. These are all classes that you studied in the classification. Okay. So Bacillariophyceae is otherwise known as diatoms or golden brown algae. They are very microscopic planktonic forms, but they are very beautiful to see under microscope. So in this class you have to study one group, that is pinnularia, one member, that is pinnularia. Okay. Now, so there are some specific features for diatoms other than that we studied in our chlorophyceae, xanthophyceae, etc. So their thallus or plant body is diploid cell, 2n in number, and their cell wall, the body is made up of cell wall, you know. The cell wall is silicified, that is very important feature, silicified, made up of calcium, silica or carbonate, or silica is the important component of its cell wall. Then storage food is also important, oil and chrysolaminarin or volutin, some special storage food is present here. Then if flagella present only a single flagella, then reproduction is by vegetative and sexual, no asexual reproduction, and sexual reproduction is by a method known as oxosore formation. These are all new terms, everything is new to you. And chlorophyll A and C present, but chlorophyll B absent, fucoxanthin is also absent. But an accessory, an extra pigment, that is brown pigment, namely diatomin present. So what are the peculiar features of this algae? That is the presence of the silicified cell wall, presence of diatomin, and the storage food materials are oil and chrysolaminarin. These are questions usually asking for multiple choice types. Okay, oil and chrysolaminarin. Clear? Okay. Now, the pinnularia systematic position is order Pennales and family Naviculoideae. Naviculoideae. Okay. Its occurrence, where can you see pinnularia? It is usually aquatic. Some are present in damp soils, wet and damp soils, but most of them are aquatic forms. Okay. Some are epizoic. You can see on parts of other organisms, especially the zooplanktons likewise. Okay. Then they are autotrophic. What do you mean by they can prepare their own food? They are cosmopolitan, universally distributed all over the world. Okay. Thallus structure, very simple thallus, but you can observe only under a microscope. When you take a 1 ml of water and put it in the slide, you can see huge number of fresh water, huge number of these planktons are moving here and there. So pinnularia may be also present in that group. Okay, they are diploid unicellular cells. They are 2 or 3 cells, sometimes may colony they form. They form colony by mucilaginous sheath. You studied one colonious form. What is that? Volvox. So cell consists of cell wall and protoplast. So there is a cell wall and protoplast. Protoplast contains all other materials including nucleus, pigments, everything. Then there are silica wall layer, outermost layer is silica. And the cell is usually like a box. You know our Petri plate. In your laboratory, you while sectioning you will get a one small glassware, that is a vessel, round vessel, where you are putting in your material for sectioning. That is a Petri plate. So it's look like a Petri plate or a box with a lid. Clear? That's it's a shape box like with a lid. Clear? Okay. So the cell wall is called a frustule. The cell wall of these diatoms are called a frustule. There are so many terms coming under this group, and usually they are all asking for examination. So cell wall is called a frustule, and the frustule is made up of, cell is made up of two equal halves that are called a valve. There are two, a box like, I already told, a box like two. You can see here a box like with two equal halves. One half, this is the first half and this is the second half, like a box. Then these halves are called valves. They are called valve. So there are outer valve and inner valve. Outer wall is epitheca. Epi means top, mugalil nartham, epi. Hypo means below, thale. So outer valve is epitheca and inner valve is hypotheca. Understood? So this is epitheca and this is, so it appear like this. This is what our hypotheca and this is our epitheca. So this is the cell, this is the cell with the epitheca and a hypotheca. Understood? So epitheca and hypotheca are the important parts and they are known as valve. Now another, there are overlapping region. You can see this region, this is the overlapping region, this is the overlapping region, two walls. That area is, in the case of epitheca, this upper side epitheca overlapping region is called epicinculum. Hypotheca overlapping region is called hypocinculum. But together you can call it as what? Girdle. So the overlapping region is termed as what? Girdle. That is very important. Overlapping region is called girdle. So you can look this algae into this side, that is girdle view. Or you can look from top side, that is what? A valve view. Clear? So there are two halves. One is epitheca, another one is hypotheca. Usually epitheca fits over hypotheca. Hypotheca is down. And the overlapping region is known as what? Girdle. Okay. So there are valve view and girdle view. What is a frustule? The cell is called a frustule. Now, so pinnularia can be seen two view under microscope. One is valve view and another one is girdle view. Okay. So what is valve view? What are structures you can see? The fine markings on the valve are called striae. Fine markings on the valve are called striae. And their markings composed of dots. They are known as punctae, and a median fissure, a structure going down the middle portion, that is known as raphe, and thickened area in the middle is central nodule, thickened area on both sides is polar nodule. See in the next slide, this is a structure. So this is a cup-like, one is valve view and another one is girdle view. You can see what is the structure. This is the outer cell wall. These are the structures of arising from the cell wall. They are known as what? Punctae. And this is a central fissure going down from top to bottom. That is what? That's known as what? Raphe. And this is the central thickened area. Central thickened area is called polar nodules. And these are the thickened area in the polar regions or apical regions. They are known as polar nodules. Okay. So cell wall, that is a frustule. And there are the epitheca and hypotheca, overlapping junction, girdle, and you can see from valve view or girdle view, one fissure going through the center, that is the raphe, and number of markings on the wall, that is punctae, and you can see there are thickenings in the center, central nodule, thickenings at the both ends, that is polar nodules. Understood? Very, very important. You have to draw the diagram and explain the structure of pinnularia. Okay. Now, protoplasm contains cytoplasm, then central vacuoles and chromatophores. Protoplasm contains cytoplasm, large central vacuole and chromatophores. Okay. Then pyrenoids are also present. Reserve food material, I already told you what are the reserve food material, fatty oils and leucosin, chrysolaminarin, etc. Jerky movement or gliding movement is present. Here flagella is absent. Now, reproduction by vegetative as well as sexual methods are there. Vegetative is simple by cell division. What is cell division? A cell divides into two. But here some peculiar feature in the diatom of pinnularia. The cell division is peculiar. So before cell division, the two halves, what are the two halves? Epitheca and hypotheca. They separate and protoplasm divides into two. So protoplasm portion move to epitheca and portion move to hypotheca. Clear? So one side of the protoplasm remains naked usually. So protoplasm all side is not completely filled. Now these epitheca and hypotheca of mother cells, they behave as epitheca of daughter cells. So you know here we can have two halves. One is epitheca and the other one is hypotheca. Clear? So while cell division, what happens? This move apart, this move apart. Okay. So this is separated and form like this, separated. Okay. So its protoplasm divides, here also protoplasm divides a small portion. Now this is the new one, this is the new one. Then now from this new cell produced from this new cell. So while forming new cell, what happens is the mother cell behave as epitheca. So this is the mother cell now. Now this behave as epitheca. This is another mother cell that behaves as epitheca. So where you can see epitheca on the top. Okay. So what happens is in the first half this is actually the epitheca. So a new cell hypotheca developed inside this. Okay. But what about the second condition? This is the actually this is a hypotheca, but it behaves like epitheca. Then what happens? The new cell formed, the new one formed inside this. Clear? So this is the way it undergo division. I will show you the picture. The new walls deposited, connecting strands developed. Both the theca, epi and hypotheca, behaves as epitheca. What happens is, on one side, where hypotheca behave as epitheca, what happens is cell become reduced in size. See, I will show you. So, this is the structure. This is the epitheca, and this is the hypotheca. Clear? So, what do you mean by epitheca and hypotheca? Epitheca is the outer part and hypotheca is the inner part. So, epitheca and hypotheca. So, what happens is first during cell division, it separates. Okay. So, epitheca here what? This is the lower portion balance. Okay. This upper portion here epitheca, this lower portion here. Now, this acts as the mother cell. What happens is always this acts as an epitheca. Always this acts as an epitheca. So, what is now forming hypotheca? Where hypotheca formed inside epitheca. Okay. So, what is here forming is new one. I will take another color. So, while forming a new one, that is always inside this. New one is inside this. What about here? New one is inside this. Understood? Okay. Now, what happens is, here again it undergo division. Here again it undergo division. Then in this division, what happens? One portion again, both behaves like epitheca. Okay. So here there is one portion like this. Here also one portion like this. Now a new one have to formed. We can take another color. So what about the new one? New one is produced inside this. Okay. Here also new one produced inside this. Okay. Now see the red color. So its one part is here. The one part is already here. That behaves like what? That behaves like a epitheca. Then what is formed inside? A new one should formed. The new one will be formed inside this. Clear? Okay. Now again what happens? Again another division continues. Again here another division continues. So here also it divides. Okay. So what happens is, see, the new one on one side it become reduced in size. Those hypotheca become epitheca. So see, usually hypotheca is small one. So epitheca formed outside to this. So again this red color act as the epitheca. Then new one produced inside this. So on one side usually the size is going on reducing. Understood? I hope you understood well. Nothing in that box like structure. ഒരു പെട്ടി പോലെ അല്ലേ, അതിൻ്റെ മുകളിൽ ഭാഗം എപ്പിത്തീക്ക, ഉള്ളിലുള്ള ഭാഗം ഹൈപ്പോത്തീക്ക. അപ്പോൾ ഡിവിഷൻ നടക്കുമ്പം എപ്പിത്തീക്കയും ഹൈപ്പോത്തീക്കയും സെപ്പറേറ്റ് ചെയ്യും. സെപ്പറേറ്റ് ചെയ്ത് കഴിഞ്ഞ് കഴിഞ്ഞാൽ പിന്നെ രണ്ടും എന്തായിട്ട് ഫങ്ഷൻ ചെയ്യും? രണ്ട് എപ്പിത്തീക്കയായിട്ടാണ് ഫങ്ഷൻ ചെയ്യുക. അപ്പോൾ ആ ഓരോന്നിലും ഉണ്ടാവുന്നത് പുതിയതായിട്ട് എന്താ പിന്നെ വേണ്ട ഭാഗം? ഹൈപ്പോത്തീക്ക അല്ലേ. അത് എപ്പിത്തീക്കയുടെ ഉള്ളിലാണ് ഫോം ചെയ്യുക. അങ്ങനെ ഡിവൈഡ് ചെയ്ത് പോകുമ്പം ഹൈപ്പോത്തീക്ക എപ്പിത്തീക്കയായിട്ട് ഫങ്ഷൻ ചെയ്ത സൈഡിൽ എപ്പോഴും എന്തായിരിക്കും? സൈസ് ചെറുതായിട്ട് പോകുന്നതായിട്ട് നമുക്ക് കാണാൻ വേണ്ടി സാധിക്കും. സി, ഇതാ ഇവിടെ ഒരു ഡയഗ്രാം. സോ ഇൻ ദിസ് യു ക്യാൻ സി ദ new development. Okay. So that's all about cell division. Now sexual reproduction, it is by oxosore formation. So oxosore formation is during the cell division, those cells become reduced in size on one side, that hypotheca act as epitheca. In that side finally reduced to size, that behaves like a oxospore. Okay. They never undergo multiplication. So production of oxosore is usually from two conjugating cells. So this is a common process. Two cells come closer to each other, and the two cells are enclosed by a mucilage cover. Then what happens is the nucleus is already diploid. So from each cell nucleus undergo meiotic division and 4 nuclei formed, and of which 2 nuclei undergo degeneration and only 2 functions. Those balance, the 2 nuclei, see here, they behave as gametes. They are formed in each cells. They are behave as the gametes. Picture I will show you. This is the first pinnularia. This is the second pinnularia. So they are covered with a common mucilage. This is the mucilage cover. What happens is nucleus, first a diploid nucleus. This is the diploid nucleus. What is diploid? 2n. Okay. Diploid nuclei undergo meiosis, 4 haplonuclei, 1, 2, 3 and 4, of which this one and this one, what happens? They undergo degeneration, and only this and this will function. Okay. Here also same. They undergo, so conjugation tube produced in between this fusion. See, fusion take place and zygote. This is the formation of what? That is the zygote. Okay. Zygote formed. So here also this between two cells of fusion, another zygote formed. Clear? Now what happens? It enlarges, elongates and become new thallus. That is the oxospore formation. So both gametes of a cell active and fuse with the gametes of other cell. Thus 2 zygotes are formed. Okay. Now zygote elongates and function as oxospore. The zygote elongates and function as an oxospore. That is covered by a layer known as perizonium. So around themselves develop new frustules and inside the perizonium. Okay. So what is a fused condition? That is zygote, that is covered by a thick wall, that is perizonium, and they become new thallus. So that's all about asexual reproduction. So asexual reproduction is by oxospore formation. When oxospore formation formed, 2 cells come closer, covered by mucilage. So nucleus is diploid. Diploid nuclei undergo meiosis and produce 4 haploid nuclei, of which 2 undergo degeneration and 2 functions. Clear? From opposite cells, a conjugation tube formed and 2 nuclei fuses, like 2 zygotes formed. Then later what happens is zygotes function as oxospore and they germinate. They covered inside a layer called perizonium. This is the sexual reproduction in pinnularia. Okay. So I hope you understood well. This is very important. Some questions may be arising. What is the structure of thallus in pinnularia, or what are the typical feature of diatoms? What is the reserve foot material in diatom? What is a frustule? What is girdle view, valve view? Then how cell division occur in pinnularia or cell division occur in diatoms? What are how oxospore formation take place, or how sexual reproduction take place in pinnularia? These are all some very important questions usually asking for examinations. So I hope you understood well. Thank you.